A lot of the times we recommend using AOPI for viability measurements simply because if you're using primary cells, you may have residual red blood debris, red blood cells as debris even post-lysis or Ficol gradients. You may also have tissue debris, you may have cellular debris, which all of these things could interfere with counting if you're using trypan blue. If you're using clean cultured cell lines, you are, of course, always welcome to use trypan blue for purified and primary cells. And the system is capable of performing both fluorescent-based viability detection methods as well as trypan blue. And today we're going to go over a little bit about GFP RFP transfection efficiency assays as well as GFP PI viability assays. So a little bit about the FOMs, which stands for fluorescent optics modules. Those are the two interchangeable and exchangeable modules that are located in the back of your instrument, and each one corresponds to a specific wavelength, works essentially as a filter for the excitation emission spectra that you are interested in using in lab. So for example, the 470, 535, 595, 660, and 695 are our fluorescent optics modules that we have available to our customers. And because of that, they really have a wide range of UV, everything from the UV-based excitation to use for DAPI and Hurst to FITC, rhodamine, Nile red, PE, as well as APC. So the entire spectrum, fluorescent spectrum, is really covered, and depending on the work that you do in your lab, you can use these fluorescent optics modules to freely interchange from one to the other. So some of the common primary cell line analysis that I wanted to share with you are PBMCs, splenocytes, stem cells, tumor cells, as I mentioned before, bone marrow cells, whole blood, cord blood. All of these are typical primary samples that are often used by our customers either before or after cryopreservation for downstream experiments, for plating to find out what is the nucleated count, what is the count of your nucleated cells in your sample, and so forth and so on. In terms of cell lines, as you can imagine, there are many, many, many different cell lines. Currently, we have counted over 1,600 different cell lines, and some, just to mention a few popular ones, are Jercats, those are the ones we'll be using today, Hex cells 293, as well as HeLa cells. So the chances are, if you're working with a cell line, we've counted, and if we have not, we'd be more than happy to work with you to help you get the most accurate and perfect and consistent count possible. So that just gives you a nice little outline of what a vision system is, what it's capable of, what kind of samples that we perform, as well as I talked to you about your options of using fluorescent optics modules that are used in different types of experiments based on your antibodies or your dyes at hand. Now let's get a little bit familiar with the vision software itself. So once you turn on your software and it's connected to your Cellometer, you will see a screen like this. So in this particular case, you're looking at a bright field image. I believe this is a PBMC image. And we have lots of different functions that are on the screen that we can talk about. The function on the left is the setup. This is where you have a drop-down menu. You can choose your assay that you're running, as well as the cell type that you're using. You can also edit these cell types and assays, but we can go into that a little bit later. The next part is sample ID. So if you, for example, as I will show you, you can save to save your data automatically. You can enter your sample ID here, as well as the dilution factor that you may be using. So very often the dilution factor is 2. As you can imagine, you can have your cell sample stained with AOPI at 1:1, and that is the dilution factor. However, you can also do, if you do any previous dilution of your sample, for example, if you're using whole blood and you would like to do a 1:10 dilution prior to staining with AOPI, you would obviously put a dilution factor there of 20 for a final and total dilution factor. You can then preview your image. This is shown in a bright field, B1. And you can preview your fluorescent images. So F1 corresponds to your green channel, and F2 corresponds to your red channel. On the right-hand side, we have image-based tools that are usable for after the image has been acquired or during preview. So on the right-hand side, as I'm showing you in the right box, you have multiple images that are collected, 4 images specifically, A, B, C, and D. And the little 1, 2, 3, and 4, each image is broken into 4 quadrants. And you are able to image and look at each individual quadrant for each cell. And I will show you that a little bit later in the presentation. You can also choose which image and which fields you're looking at, whether you're looking at the bright field, whether you're looking at a fluorescent field, or whether you're looking at fluorescent green or red. So you can easily use these little boxes to check from one field to the other. You can also check these boxes and look at your counted cells. You can also combine the 2 images, which overlays the red channel with the green channel to show you the total fluorescence that is acquired. On the top left corner over here are is the results template, Cellometer calculator, which allows you to calculate your percent of cells that you may need for plating or for densities, as well as cell size histogram. So this gives you a basic outlook of what our software looks like, as well as how to use it. So as I had mentioned, number 1 is the result template. So after your sample is finished counting, you automatically get a pop-up window, which is our result template. And the result template has several important features. First, it tells you what is the assay that you're using, as well as what kind of cell type that you used. It will have a timestamp of when this assay was performed, as well as the date. It will tell you your sample that you typed into your sample ID, as well as the dilution factor for each individual sample. The results show count of fluorescence 1 with the green cells, fluorescence 2, which are your red cells, or acridine orange or propylium iodide. It will show you the concentration of each population. It will also show you the mean diameter of each population. And then the viability is automatically calculated. In this particular case, as you can see, the viability was 94.2% for this PBMC sample. We also report the mean fluorescent intensity for each channel. 21.7 counts was for the acridine orange channel, and 21.1 was for the propylium iodide channel. So the question is how to perform an AOPI viability assay. And so I would like to show you a brief video that talks about this assay.
It's most suitable for combinations such as white cell and green cells and mixed cells, which contain debris and
Let me pause here for a second and increase the volume. The volume seems to be a little low.
Dual fluorescence AOPI assay for cell concentration and viability. This assay is most suitable for standard cell samples, such as white blood cells, glenocytes, and lymphocytes, which contain debris and unwanted non-nucleated cell types, such as red blood cells, acridine orange, and propylium iodide. Are nucleic acid binding dyes. They produce strong fluorescence after binding to DNA. AO is permeable to both live and dead cells. PI is only permeable to dead cells. Cells are mixed with both acridine orange and propylium iodide. Channel 1 of the cellometer is configured with an optical module with blue excitation and green emission. for a live cell, AO enters the cell membrane and binds to its DNA. Under blue excitation, strong green fluorescence is emitted and detected. PI is excluded from a live cell. Both AO and PI enter a dead cell through the damaged cell membrane and bind to its DNA. Under blue excitation, the green fluorescence by AO is absorbed by PI, producing an extremely weak green signal. Channel 2 of the cellometer has green excitation to detect red emission. Live cells exclude PI. Without its DNA binding, there is no detectable red light from live cells. For a dead cell, PI enters the damaged cell membrane and binds to its DNA, producing a very strong red fluorescence. To perform a cellometer AO-PI assay, load stained cells into a counting chamber. Insert the chamber into cellometer. Cellometer automatically performs cell image acquisition and imaging analysis. Typical assay time is less than 60 seconds. Cellometer can also perform many other dual fluorescent viability assays. Explore our website at Nexcelom.com for more information.
Great. So I hope this gives you a little bit of a better understanding specifically of how the AOPI assay works. So our typical protocol, as was alluded to in the video, is we take 20 microliters of the cell sample, which is really a small amount. We mix it at 1:1, 20 microliters with acridine orange or propydium iodide in the tube. And there is no staining or incubation period. It's virtually instantaneous. You pipette the 20 microliters of your sample directly into the disposable counting chamber. You can load one side, as you can see in this image. Either side can be loaded. So once the slide and sample is loaded into the slide, you insert the slide into the cellometer, and then you choose your assay from the drop-down menu. So I will show you that in a second as well. You simply click Count and acquire an image and view results. So let me actually go ahead and show you a live demo. I have a sample here. Here's my Cellometer Vision. So as you can see from here, I have currently selected a bright field assay. So as I have stained the sample, I took 20 microliters of my cells. I took 20 microliters of my dye, mixed them 1:1, loaded into the cellometer, and now I will choose my assay. For this particular assay, I'm going to perform an AOPI assay. I can set my sample ID. So my sample ID, these are Jerkat cells, Viability. Okay, so it's shown down here with the sample ID. I can set my dilution factor, and I can also preview. So I can preview F1, so preview bright field. So this shows you my cells, as you can see, as I'm focusing through the field, through the cells up and down. It's really nice to have a clean, crisp membrane of these nice, beautiful, bright, live cells. I can also stop the bright field preview, and I can preview these same cells, same population, in a fluorescence image. So I'm previewing these images now in the green channel. I will again stop preview, and I can preview the same images in the fluorescent channel. So we will now go ahead and acquire these images. So once again, I would like to make sure that the plane of field that I have selected is good, and I simply press Count. So when I press Count, as you can see, you will circle through the images. On the bottom, it's telling you exactly which image is being acquired: fluorescence, bright field A, and bright field B. So the reason is you might wonder why we select the second bright field image. Because this is a dual fluorescence assay, a bright field image is collected for each the green and the red channel. Although we do all of our focusing only in the B1 channel. So there are 4 sets of images that are automatically being collected: image A, image B, image C, and image D. So this image D is almost finished being collected. It takes literally several seconds to collect all 4 images, and then counting is extremely fast and almost instantaneous. So once the counting is complete, this is the result template that I showed you previously. It shows you your assay. It shows you your cell type that you use. In this case, it's immune cells, the low RBC. These are the cell type that I created. The sample ID was a Cat cell viability that I typed in. This is my dilution factor, and I performed this experiment today. So it also gives me total count of my cells, bright field number of cells, total fluorescence, green, live which are AO positive, and dead, which is PI positive. It also gives me a concentration breakdown of each population as well as the mean diameter. So the viability for this sample was 47.3%. Don't be alarmed, my cells are healthy. I chose to mix them on purpose. 50% live cells roughly and 50% dead cells were mixed to acquire this viability. And so now that we have acquired these images, we can go ahead and take a look at all these little functions that I had shown you previously about our cells. So we can look at, for example, a bright field image. So as I said, there are 4 images that were taken: image A, image B, image C, and image D. We can choose to zoom in any one of these 4 quadrants. So each image is broken into 4 quadrants. We can also choose to zoom in even further if we want to really look at our cell morphology or look at our cell population much more closely. We can also look at our images in the F1. These are live positive cells. We can also look at our images that are PI positive. These are our dead cells. Down below, we can click Counted. So a green circle represents counted cells. So as you can see here, all cells are nicely counted, and each dead cell is identified. We can similarly do the same thing for the AO positive cells. And as you can see, as I go through these different images, all of our AO positive cells are also nicely counted. I can also click Combine. Combined overlays the 2 images, the red with the green. A couple of things I wanted to bring to your attention here is the declustering capabilities of this software. As you can see, there are several areas right here where in the bright field, these cells are fairly close together. As a matter of fact, they're touching each other. But when we're looking at combined total counted images, you can see that each individual cell in each case is counted. So this is really the strength of this software, where we have a specific algorithm that is designed here in-house that assures you of accurate counts all the time. So once again, we can go to this result template. I will show you a little bit later on about some of these functions, how to view and export your data. This is also the sample adjust calculator and the cell diameter histogram. The cell diameter histogram essentially shows the population of all of your cells here, as well as the distribution of your AO and your PI populations. It shows you the mean diameter of your distributions. Okay? So let's go back to the presentation. And so this is how we have just performed a live AOPI viability assay. And now I will show you a little bit about GFP RFP transfection efficiency assay and how that works. So once again, you would go down and you would select your assay from your assay drop-down menu. So I would choose the GFP transfection. I'm going to load an image, so that's the nice thing about this. You can always go back and load any image that you want. These images can be saved automatically for data analysis at any point in time. Once the image is loaded or the cells are counted, as you can see, the count is extremely fast. We are looking at, once again, this is the assay that we just ran. Sample ID, these are the cells that were used, HCC 116s. The dilution factor was 1 because we didn't dilute it. We did not stain it with any reagents. These are GFP expressing cells. You have a total cell count and the number of GFP positive cells. Once again, the concentration and the mean diameter of each population, as well as the percent of GFP positive, which is 37.9%. We can go ahead and once again closely examine each population. We can choose counted, and we can look at each cell that is counted as outlined in green. And we can look at the red. We can look at the green channel. So as you see here, this is slightly different from the AOPI. Since this is a GFP transfection assay, only one color is being used, the GFP, the green channel. And so in here, the green circles represent counted cells, or GFP positive cells, and the red circles represent the non-GFP positive cells. These are the cells that, as you can see, as I toggle in between, for example, these two cells right here, or these four, we can look at them. They are counted in bright field, and this is also shown in the fluorescent channel. You can also include uncounted. So I will also go through the GFP and PI. Let's first do the RFP as promised. So once again, you would go down to your drop-down menu and choose RFP transfection rate. I would load my RFP image. And as you can see, these cells have a very strong expression of RFP. 42.2% of these cells are RFP positive. Once again, you can click on the counted image to assure yourself that all your cells of interest are indeed counted. You can go in between and go through different fields of view. And you can then click on the RFP. As you can see, very strong RFP signal in this particular case. All of the cells that are counted RFP positive are outlined in green, and all other cells are outlined in red. The nice thing about this that I will mention very briefly is that we do have settings called the fluorescent threshold settings. And so if a researcher is expecting to have a very high expression of RFP or GFP, and then you can have a second population where you have a low expression of RFP and GFP. So in this particular image, as you can see, there are some cells that are RFP dim, and there are some cells that are RFP bright. Based on those fluorescent thresholds, we can determine what population we would like to examine and which population we would like to look at. And so if we believe that our high RFP positive cells are their population of interest, we can simply increase our fluorescent threshold. Therefore, we only count RFP positive cells, RFP strong, RFP high positive cells, and that's what I've done here. And so finally, we can look at a GFP PI. This is very useful for those that are looking to do viability at the same time as looking at your GFP positive population. So the image is loaded, and it is counted within seconds. We can look at, once again, I will come back to this template and show you all the details in the template in the results section. Once again, you have these, I believe, are mouse stem cells that we're looking at here. So once again, we can go through and look at the bright field images. Here are our GFP positive cells. They are in green. And since we do have a PI channel here, this is a dual stain GFP PI. We also have PI positive cells. The overall red circles that you're looking at are the cells that were counted in bright field. And so you can imagine that you can have a population of GFP positive cells, you have a population of PI positive cells, and you can have a very small population of GFP positive cells that are either dying or have a compromised membrane, and that would be dual positive. So if we go back and look at the result template, in our results, we have F1 channel, which is the green number of cells, F2. We have a total number of cells counted in the bright field channel. We have a concentration for each as well as the mean diameter. So here are the ratios that we report. We report percent of GFP positive cells, percent of PI positive cells, so these are your dead cell population, and percent of dual positive, these are GFP and PI. You can also simply calculate the viability by subtracting 100% of your cells from your PI positive cells, so 100 minus 28. So in this particular case, your viability is 72%. Okay? So I would like to also just quickly talk about some data management features, specifically the sample adjustment calculator, dilution factor, histograms that I had previously shown with the AOPI, as well as auto-saving and printing options. So I can go back and I will actually show you all of those features on our live sample to give you a better feel as to how things can be run and organized. We have up here, we have four buttons. You can click on the option button and save options. So these are particular option details that we can choose to do. We can have auto-saving your data files, auto-saving an export image Nexcelom data package, or auto-save sample images. You can have raw samples, GFP images, save counted images, and save combined images. The one thing that I will stress, by far the most important thing, is to save your raw images. Raw images can be recounted, reanalyzed. If you have issues with your cellometer, or you are suspecting that your counts aren't exactly what you want them to be and you need them tweaked, you need parameters changed, these are also the images that can be sent back to us, to Nexcelom, for us to look at, to analyze, to troubleshoot. So without the saved raw images, it's very difficult to reprocess your samples, or actually it's impossible to reprocess your samples. So I would like to also show you the autosave function. And the way you would do this, you would click autosave, and you would choose where you would like that file and data to exist. So once you've chosen that, you can simply choose a name. So for example, I will write today's webinar. Okay? And so that all of my images that I acquire during this live example, they will automatically be saved. I have also checked Save Auto Sample. Okay? So I will set my sample ID. So this is Jerkat Cells 2, Viability. Save. I will once again preview my sample, as I have done in the past, and I will count. So once again, the cellometer takes 4 images. It takes 4 sets of images, as you say, a bright field, a fluorescent 1, and a fluorescent 2 image for all of them. And it will automatically show your data results after the imaging is finished, as well as report your percent viability for this particular sample. So it's now on image D. Finishing up, we set our dilution factor to 2. I used my cell sample and I diluted it. I stained it 1:1 with acridine orange and propydium iodide. And this is the counting result template that is shown. So since we have automatically saved our data, we can click on the file that says View Data File. And as we did that, we have our data that we just set. We have the date, we have the sample ID name, we have the channel that was acquired, F1 or F2, we have the cell count for each channel, the dilution factor, the concentration. This was a viability experiment, and you have a result, 47.1% viability with a mean diameter. This can be directly sent to a print or exported to an Excel sheet. So you do not have to record after every count. You don't have to manually record any numbers. Everything is automatically exported into this template and to Excel. We can also show you the intensity distribution. So this is the mean fluorescent intensity for the red channel PI as well as acridine orange, as well as the fluorescent intensity for each one of the cells that was collected here on the left as I scroll through. We can also show you the size distribution. So this is, as I talked about, cell size histogram. This is the size distribution for both population, for the PI and the acridine orange. It shows you the diameter of each individual cell that's collected, as well as the range of cells and the number of cells that were of that size. For example, 10.2, there's 131 cells that fall within that parameter. So these are the buttons that I had shown you before. So this is the sample adjustment calculator. If you have an original volume, say of 1 mil, and you have a target concentration of 2 million cells per mil, for example, and you can apply the change, and it will tell you that you need to spin down and add 0.72 mils of cells in order to reach the concentration of interest. It's a really neat little feature that allows you to play with concentrations and cell numbers if you need exact numbers for counting. So this is essentially one of the concluding parts of our webinar, but I will go back and finish up. So we have a couple of other Cellometer training webinar topics that are coming up. We have a Cellometer K2, AOPI, and hepatocyte viability. We have how to run apoptosis and cell cycle-based assays on Cellometer Vision CBA with FCS Express, how to customize cell assay parameters, how to select your best viability method for your sample. So if you're working with some complicated sample and you're wondering whether you should be using Trypan Blue or AOPI or some other method, topic of your input, please give us feedback at any time. We're always interested in hearing from you. So at this point, I will conclude my seminar, and we will take any questions. Thank you for your time.
Thank you, Dimitri. That was great. I'm pretty non-scientific here in the office, and even I learned a few things with that, so thank you very much for your time. One of the things I will just reiterate, as Dimitri had mentioned, we are constantly looking on ways to improve the way that you're using our technology in your lab. So if you have, or you and your lab team have a specific topic of interest that you would like to see as a webinar, we can do a private webinar for you and your team. Or if it's a general idea that you would like to have, we can do a monthly training like this as well. So always feel free to reach out. You can reach us at support@nexcelom.com. Do we have any questions today? I had one, personally. When we were looking at the dual, where we wanted to see the PI and the RFP, in our report it said the total viability is you take your 100% minus your PI viability. If the PI viability is already calculated, why doesn't the program already do that math?
That's a great question. So there are certain parameters that are pre-programmed into the software, and we are coming out with, I believe, new versions of the software all the time. And particular programming features, when you're doing a dual fluorescence assay, is usually meant for example, for AOPI, where you're counting viability. But in this particular case, the counting is done a little bit differently. For AOPI, all of the counting is done in the fluorescence channel only. No bright field cells are counted. This is due to the fact that it may be very difficult or impossible to count bright field cells in your clinical samples or your primary samples that may contain a lot of debris. therefore, counting in bright field is not optimal. So you might be able to you might be picking up red blood cells or other contaminants. So all the counting is done in the fluorescent channels. In this particular assay, as you've noticed, we counted bright field cells. We also counted the fluorescent 1 and the fluorescent 2 channel. So simply the software at this particular time does not have a direct formula that is automatically represented for the viability, since the assay is actually meant to represent GFP positive cells, not necessarily a viability assay. You can also choose to do a viability assay, AOPI assay, on your sample separately. But if you're interested in a quick method to determine your overall viability of your GFP expressing cells or your GFP expressing population, that's just a simple way, an additional way that you can simply do it without having to perform a secondary assay.
Thanks. That was really helpful. Do we have any other questions today?
So the other thing I wanted to mention is that this PowerPoint will be available after this presentation for you to view again, to listen to again, to share with your lab mates. You may simply just have to log back in to the same link, and then it will be available.
We will also have the complete slide presentation available as a PDF. So if you or any of your lab members are interested in having a PDF version of this, of the PowerPoint presentation, we can certainly get that to you. Again, you can just email us at support@nexcelom.com and be happy to send that out to you.
Yep, and one other thing to mention, if you do have any questions, you can contact Ben. So Ben is our technical support specialist, and he will be more than happy to answer any questions or problems or troubleshooting that you may have with your assay or with your instruments. He also works closely with the R&D team, so if he has any further questions, he relates them to us. So his email is bparadise@nexcelom.com, and you can contact him with any technical support or technical questions as well.
Well, I want to thank everybody for joining us today. I hope that you got a lot out of this. And stay tuned. We are going to be conducting Cellometer user training webinars every month on the 4th Friday of the month, and invites will be sent out accordingly. Again, feel free to get in touch with us if you have an idea for a topic you would like covered. I'd like to thank again our presenter, Dimitri, today. Thank you very much. And always feel free to get in touch with us if you have any questions. Thank you.
Thank you for joining us. Take care.